Session 1 of cells, transcription. Transcription happens after the DNA in the nucleolus needs to be transcribed. It's the process where genetic information encoded in the DNA is copied onto a strand of RNA called messenger RNA. First of all, the promoter region initiates transcription by RNA polymerase binding to a start base, and this start base is AUG. Elongation. RNAP reads a base triplet at a time and makes a complementary RNA strand. Elongation. RNAP reads the base triplet. Termination. So I'll do elongation again. Elongation. RNAP reads a base triplet at a time and makes a complementary RNA strand, mRNA, from both intron and exon sequences. DNA is only unwrapped at the site of transcription. Termination. RNAP stops transcribing when it reaches a stop base triplet. And it says here a stop base triplet might be TAA, TAG or TGA, TAA, TAG or TGA. So we've just read about transcription, and that's a process for transcription. Once the RNA is complete, begins the process of translation, which is the process of reading the mRNA nucleotide sequence to determine the amino acid sequence. For the initiation, the mRNA initiation. mRNA with a start codon of AUG is first read by the ribosome with the antisense tRNA anticodon. Initiation, this is the translation initiation. mRNA with the start codon, AUG, is first read by the ribosome with an antisense rTNA codon. Elongation. mRNA is now a template for ribosome to keep translating codons into anticodons. This will build a polypeptide chain that will become proteins. Elongation. mRNA is now a template for ribosome to keep translating codons into anticodons on tRNA. Termination. Once ribosome encounters a stop codon on mRNA, a corresponding tRNA signals the release of polypeptide chain from the ribosome. Once ribosome encounters a stop codon on mRNA, corresponding tRNA signals release of polypeptide chain from the ribosome. First of all, A. The initiation tRNA carries the amino acid. B. C. D. The first for D, this most important one, and I think the only important one, the first tRNA is released and the ribosome moves one codon to the right, which translocates the dipeptide to the P site and the tRNA to the A site. That's session 1. Session 2, integral proteins. Extend into and across the entire lipid bilayer. Peripheral proteins are found at the inner or outer surface of the membrane and can be stripped away without disturbing membrane integrity. The ion channel is an integral protein. The ion channel is an integral protein. It forms a pore which a specific ion can flow to get across the membrane. The cell identity marker, which is also called a glycoprotein, is an integral and peripheral protein. It distinguishes your cells from anyone else's. This is an example of a ligand receptor, and it's an integral protein. It recognises specific ligands and alters cell functions in some way. For example, the hormone in kidney changes water permeability. Ligand recognises specific ligand and alters cell's function. The integral, another integral is the linker. And a linker anchors filaments inside and outside the plasma membrane. Structural stability. This is integral and peripheral. Most are integral. The peripheral ones link one cell to another cell. Another one is substrate. And this is called an enzyme. An enzyme is also integral and peripheral. Enzyme is integral and peripheral. It catalyses reaction inside or outside the cell, depending on which direction the active site faces. An enzyme catalyses reaction inside or outside a cell, depending on which direction the active site faces. Lactase lactase in intestinal cells as an example. The carrier is also an integral protein. It transports a specific substance across the membrane by undergoing a change in shape. The carrier transports a specific substance across the membrane by undergoing a change in shape. So the only ones that are both are the cell identity marker, which differentiates your cells from anyone else's except an identical twin, and an enzyme. That's session 2. Phospholipids is what cells are made of. Concentration gradient is a difference in the concentration of a molecule between two sides. For example, oxygen and sodium ions are more concentrated outside the cell. Electrical gradient, difference in electrical charges across the bilayer. And we call with the lipid bilayers amphipathic, which means polar head and non-polar tails. To recap on mitosis, we have the G1 phase.
Ready, Dad.
And this whole process results in 2 daughter cells, each having the same number and kind of chromosomes as the parent nucleus. This is different to meiosis, where the process is genetic diversity, so they're not the same. Interphase. Cell is initiated by duplication of its genetic material. G1. Cell metabolic rate increases. S phase. DNA replication and chromosome condensing. G1 phase. Cell metabolic rate increases. S phase. DNA replication and chromosome condensing. G2 phase, conclusion of metabolic increase. G1, cell metabolic increase. S phase, DNA replication and chromosome condensing. G2 phase, conclusion of metabolic increase. Prophase, metaphase, anaphase, telophase. Prophase, metaphase, anaphase, telophase. And in the interphase, entry point into mitotic division. So the first one is prophase. Early prophase shortens into visible chromosomes. Late prophase, nucleolus and nuclear envelope disappears. Late prophase, nuclear envelope disappears. Each centrosome starts to move towards opposite ends. Each centrosome starts to move towards opposite ends of the mitotic spindle. Metaphase, they line up along the metaphase plate. Anaphase, early. Anaphase is characterized by splitting and separating of centromeres. And the late phase is characterized by the movement of the two chromatids of each pair towards opposite poles of the cell. The movement of the two sister V-shaped chromatids of each pair toward opposite sides of the cell. Telophase. The identical sets of chromosomes uncoil and revert to their thread-like form. The nuclear envelope forms and a new nucleoli appear. Then we have the cytokinesis, which is the division, where the cleavage furrow is completely broken. Then we go on to cell junctions. These are the different types of cell junctions. We start off with tight junction, and the function is a watertight seal. The structure is strands of transmembrane proteins. It can be found between cells that line the GI and bladder. Function of tight junction: watertight seal between cells, can be found between cells that line GI and bladder, and the structure is strands of transmembrane proteins. Some cell junctions form fluid-tight seals, others anchor cells together, and others act as channels. So the three functions of cell junctions are fluid-tight seals, which we just saw with the tight junctions, anchor cells, which is the hemidesomes, and the channels. What are the three functions of cell junctions? Fluid-tight seals anchor cells together or to extracellular material and act as channels. Adherens junctions. The structure is plaque forms a dense layer of proteins inside the cell. Integral membrane proteins connect the membranes of the two cells. It holds epithelial cells together. It can be found between cells that line the GI, adherens junctions, GI. Tight junctions, GI. No leaking in GI. Gap junctions. The structure is a tiny space crossed by protein channels called connexons, and they are fluid-filled tunnels. The function is cell communication with ions and small molecules. It can be found in the heart and smooth muscles of the gut. Muscle and nerve impulses spread from cell to cell because they travel along the fluid-filled tunnels. desmosomes, hemidesmosomes connect the cell to the cell membrane, cell to the cell basement, to the underlying basement membrane, and uses integrins, not coherins. The function is cell to membrane stability, and it can be found in any cell attached to cell membrane. Plaque, transmembrane glycoprotein, which is integrin. Plasma membrane, basement membrane. Desmosomes. Similar to adherens junction, but connects to the intermediate filaments within the cytoplasm. Similar to structure to adherens, but it connects to the intermediate filaments within the cytoplasm. Function is cell-to-cell stability. It can be found in the skin and cardiac muscle. Skin and cardiac muscle. Then we go on to the classification of epithelium.